Okay, that's it. Okay. We good? Yeah. This is fun waiting. You don't know what's gonna happen.
How long is it? Are you gonna go alone?
She got in last night and picked her up for last night and just find out something.
Oh, good, okay.
Yeah. Yeah, she's gonna. We came through the halls last night, girl, and there was. So they had to. I wrote it down. But then the interstate, they closed the interstate, so we just. The end. Oh, and then as we came back around the dolls, they like for 10 minutes.
Yeah, I'll put it with myself.
But they were like, they were looking yeah, so.
I'm good very much.
Okay. Oh. Yeah.
John is the one
Yeah. Oh, that's good. So if you accept that. Yeah. When did you say?
Yeah, I'm your fine.
Yeah. I'm sure.
It's okay, yeah. Yeah,
Yeah, I'm sorry. Yeah.
Well,
I just thought you're like, my friend actually. Yeah.
There's no question.
I have some wedding
Yeah, he took you out there. Today, I'm like,
Oh, it's good. We were actually going through the calendar. I mean, yeah. Yes. Okay.
Okay, how's the volume on this? All right. All right, well, we can get started. I'm delighted to introduce Katie McHugh, who's here for her PhD defense today. So what can I say about Katie? Katie has had to navigate some challenges, I think it's fair to say, along her graduate journey. For one thing, she's actually the first Birchlab PhD student. So she's had to deal with an unseasoned advisor. So that's one challenge. Another challenge is that she started here in IB in fall of 2019. And so she got here. We designed all kinds of super ambitious experiments immediately, like you do if you have an unseasoned advisor. And then we were all ready to launch these experiments, which involved like quite a bit of travel and precise timing of experiments. And then it was March 2020, and of course everything got shut down. And so that could be considered a crisis to some, but I was, I observed Katie over the next weeks, months, and really years, grow into an adaptive, a thoughtful, and a resilient scientist. And it was such a pleasure to watch that growth. And at the end of the day, we did all the experiments that we set out to do. And I think that they ended up even higher quality because we had to slow down, plan more carefully, think, 5 steps ahead of ourselves instead of just, 1 or 2. And so Katie, completed everything that we wanted to get done and more, right? And so today she's going to tell you about all of those results that she's curated over the last few years. And I just have to say that she's really set the bar as far as, certainly as a first PhD student, I now see what an excellent grad student can do. And all of her peers who have come after, who are grad students in the lab now, have had her example to look up to. And for that, I'll always be grateful to Katie. And somehow she even managed to achieve excellence in teaching, outreach, and even run a few marathons along the way while she was doing all of it. But for me, that sounds like a lot. So anyway, all that is to say that I'm so proud of Katie, and I'm thrilled that she's here to present the culmination of her research, her PhD research to you today. And it's quite bittersweet, to say goodbye as she's fledging, but I know it's not goodbye for reals. We still, we'll continue to collaborate and finish up some projects and get them ready for publication. So with all that being said, I'm going to hand it over to Katie.
Thanks, Molly. Sorry, give me just a second to make sure I have all of my things. Okay, we're good. All right. Yeah, thank you all so much for being here today. It means a lot to see so many of you here. I'm really excited to tell you about the research I've been working on for the past 6 years, looking at the evolutionary implications of age-related fitness traits in the yeast Saccharomyces cerevisiae. All right. So as I'm sure you've all observed, aging is highly pervasive across the tree of life. If an organism lives long enough, it's pretty much inevitable that it will eventually begin to show signs of aging. And so from shorter-lived species like the mayfly and the goby fish to longer-lived species like the Greenland shark and glass sponges, we see an incredible amount of diversity in both the timing and the phenotypes that are associated with the onset of aging, both between species, but also within them. And so I'm really interested in trying to kind of understand how this variation is both maintained and shaped within populations, and how we may even be able to manipulate it. But to be able to understand the variation that contributes to aging, we first need to understand kind of the underlying causes of aging. And so from a more kind of proximate or mechanistic standpoint, things like genomic instability, loss of proteostasis, and deregulated nutrient sensing are typically associated with cells that are at advanced age. And they tend to kind of indicate that there's some sort of breakdown in processes happening in these cells that's leading to them to no longer be able to maintain themselves, and age-associated damage can accumulate within the cells. However, these more mechanistic causes can't get at kind of a broader explanation of why aging exists at all. Why can't our cells just indefinitely maintain themselves, allowing us to remain immortal? And to answer this question, we need to turn to evolutionary theory, and specifically the evolutionary theory of aging. And so we know that the force of natural selection acts on populations rather than individuals. And so, sorry, I forgot it. And so detrimental mutations that take effect very early in life are likely to have large impacts on fitness because they're very likely to impact the ability of organisms to pass on their genes to the next generation. While detrimental mutations that take effect very late in life are unlikely to impact fitness because most organisms will have either already reproduced or died by the time these mutations take effect. Because of this, late life mutations are often essentially invisible to the force of natural selection, which allows detrimental mutations to build up in the genome, which we call mutation accumulation. Similarly, mutations that have both beneficial early life effects and detrimental late life effects will actually be selected for under natural selection because that force of selection is so much stronger early in life, which we call antagonistic biotropy. And so this can lead to trade-offs where we see improved fitness traits early in life at the expense of fitness late in life. And additionally, together, these kind of help to explain why it's so difficult, if not impossible, for populations to escape the impacts of aging, because there's just essentially no real force that's acting against it. However, under evolutionary theory, it is possible for us to delay aging. So if we're able to delay the age of first reproduction within a population, then the force of natural selection should remain stronger longer, effectively delaying the onset of aging. And this isn't just theory either. It's also been shown empirically. In the 1980s, there's kind of a landmark study done in populations of the fruit fly, Drosophila melanogaster, that showed that selection on delayed reproduction can essentially double the average age within these populations. And this result has been repeated in several other lines of fruit flies. And we frequently observe trade-offs that we would expect under evolutionary theory, although what these trade-offs are tends to vary between populations. However, only a few studies have attempted to look at types of evolved longevity in species other than Drosophila melanogaster, and the only genomic studies that we've observed have been in fruit flies. And even though these, while exciting, are generally kind of under-replicated for us to draw any really strong conclusions about what they're saying. And so if you're interested, Molly and I wrote a review paper on this a couple of years ago that kind of outlines some of the different selection regimes you can use to select on aging and longevity, trade-offs that we frequently observe, kind of pros and cons of different organismal systems that have been used for this type of work, and also some of the genomic weaknesses that we've seen. But one of the main takeaways, so I'll give you a sneak peek here, is that the yeast Saccharomyces cerevisiae is a really exciting potential model system to try to do this work. And so throughout the rest of my talk, I'm going to talk about why this is, and we're essentially going to go through and try to see if we actually can use yeast to interrogate the genomic basis of longevity using similar methods. Oh, sorry. And so yeast is a really great model for studying the evolution of aging. There's two ways that we can measure aging in yeast. They do age, kind of prerequisite, right? So the first is chronological aging, which is just the amount of time a cell can survive without reproducing. And the second is replicative aging, or the amount of asexual divisions a cell can undergo. And so you can see right here, this is a mother cell that is forming a bud. So the cell is currently undergoing asexual reproduction. And during asexual reproduction, damaged components of the cell are maintained within the mother cell, while all new components are synthesized for the bud. So in this way, we essentially get two cells that are pretty much genetically identical, but the damage is sequestered into the mother cell. And so in this way, replicatively aged yeast cells have been shown to be very similar to other aged cells in systems like humans, and it's considered a good system for studying aging in the cell protocol. Another really useful feature of replicative lifespan is that after each division, we get these scars, which are called bud scars, that form at the division site. And we can actually stain these with a stain called WGA, or wheat germ agglutinin, which fluoresces. And then we can perform fluorescence imaging in order to actually count the number of bud scars on a cell to tell us how old that cell is. And in addition, we have another method that we use called fluorescence activated cell sorting, or FACS. And we can use cell sorting to separate large cultures of yeast into cohorts of young cells that have few bud scars and low fluorescence, and aged cells that have more bud scars and high fluorescence. And so I'll use this method of cell sorting throughout all three of the experiments I'm going to talk about today. And in addition to asexual division, yeast also reproduce sexually. And so they do this through a process called sporulation, which is essentially the same as meiosis in other eukaryotes, where we start with a diploid parent cell that undergoes 2 rounds of division to form 4 haploid daughter cells, which we call spores in yeast. These spores are packaged within an ascus, which looks kind of like that. And so we can kind of load these cultures of sporulated cells onto a chemocytometer to count them under the microscope. And we can get an estimate of what we call sporulation efficiency, or just the proportion of cells in a population that has actually undergone this process to sporulate. And so that can give us an idea when we're looking for evidence of reproductive trade-offs of kind of this relationship between our sporulation efficiencies and our different populations. And so this is what that actually looks like under the microscope. You can see kind of these bubbles are the spores, whereas the cell down in the corner has not sporulated. Okay. And so now that I've kind of walked through some of the theory and the reasons that aging are a pretty good model for this type of work, I want to kind of bring us back to our broad question that we're trying to investigate today, which is how does evolution really shape variation in aging and lifespan? And so I'm going to walk through 3 experiments that I've done for my dissertation that have aimed to kind of broadly investigate this question. And so in the first experiment, I'll be attempting to identify an age-associated evolutionary trade-off in populations of yeast, which we would expect to observe under the evolutionary period aging. In the second experiment, I'll detect heritable variation underlying this age-associated phenotypic variation we see. And in the third, I'll attempt to experimentally evolve populations with delayed aging. And so to start out, we will try to identify these age-associated trade-offs. And so in this experiment, I'm using 5 diverse populations of yeast. So 4 of these are isogenic populations that originate from diverse ecological and geographic backgrounds. So YPS 128 is a North American strain from an oak forest, Y12 is a Japanese sake strain, and DBVPG 6044 is West African palm wine strain, and then DBVPG 6765 is European strain. And so while these 4 strains have a large amount of genetic diversity from one another, within each population, there's actually very little, if any, genetic diversity. So that's why they're called isogenic strains. And so each cell within these isogenic populations is essentially the same as every other cell. The 5th population we're using is called the forest population. And so it's called the recombinant population because we created it in the lab by crossing our 4 isogenic strains together. And so this population is diverse not only from the 4 isogenic strains, but there's also a lot of genetic diversity within the cells within our forest population. And this population has also undergone recent sexual recombination to kind of continue to shuffle that gene. And so we take these 5 different populations, and for each replicate, we can age our cells to generate a wide distribution of cell ages, stain the bud scars, and then we can sort them using fluorescence activated cell sorting into low fluorescence fractions and high fluorescence fractions. We then perform fluorescence imaging to count the number of bud scars in each fraction to make sure that we're actually getting aged versus young cells in our different fractions. And then we perform sporulation efficiency estimates to look for evidence of a reproductive trade-off between our aged and young cohorts. And so this is the data for the bud scar counts. You can see in this figure, there are 5 panels, 1 for each population, with our genetically diverse forest population on the far left here. And within each panel, on the left-hand side, we show all of our young replicates with a line connecting them to their paired aged replicate. And across all 5 populations, we can see that there's a statistically significant increase in bud scars in our young cohorts compared to in our aged cohorts compared to our young cohorts. And so this seems to be confirming that our sorting is working and we actually are getting these older cells in our aged cohorts. And so next we moved to look at, oh, I'm sorry, I'm ahead of myself. But unfortunately, 2 of the populations that we were using for this analysis, we were unable to get to sporulate under the conditions of our experiment. They do sporulate, and we've gotten them to sporulate previously, but there's kind of some specific methodological constraints to cell sorting that seem to be kind of incompatible with sporulation constraints. So unfortunately, we dropped these 2 from the rest of this chapter, and we're just going to continue on with the 4S and then our other 2 isogenic populations for the rest of this experiment. And so these are our sporulation efficiency estimates. Once again, our forest population is on the far left here, and our 2 isogenic populations are on the right. And what you can see here is that the proportion of cells successfully sporulated was statistically different in this 4S panel, but we don't see a statistically significant difference in these other 2 panels. And there's a few possible explanations for this. It could be that we see a trade-off in a different phenotype and not the one that we were assaying. It could be that there's just a lot of variation that made it difficult to kind of detect a change, but we were able to detect a change for us. But maybe the most interesting possible explanation is that there's actually a lot of difference in the life history of these populations, right? We kind of mentioned that earlier, that these isogenic populations have very little genetic diversity and they primarily reproduce asexually, whereas our forest population has a lot of genetic diversity and undergoes frequent sexual recombination. And so there's a lot more opportunity for natural selection to act on our 4S population compared to our other two populations. And so it's possible that the evolutionary theory of aging just doesn't really apply to asexual populations in the same way that it does to sexual populations. And so because of this, we're going to focus just on this 4S population for the rest of the talk, where we know we have this sexually reproducing population that shows this reproductive trade-off associated with age. And so now we're going to move on to the second part of our experiment, where we're going to see if we can detect heritable variation that might underlie this variation that we observe in age-associated traits. And so we know that aging is a highly polygenic trait, meaning that there are a number of different genes that contribute to the phenotypes that we observe. And in addition to there just being a lot of genes that contribute to aging, within these genes, there are different alleles or different versions of the genes that can further combine in different combinations and lead to variable phenotypes. And while the genomics of aging is very well studied, many of these studies only use isogenic systems. So that's like those yeast from the previous chapter that don't have much genetic diversity. And so studies in these isogenic populations can be really useful, but they're also going to miss some of the diversity that's present because we don't get to see these different combinations of alleles in different genetic backgrounds. Additionally, of the studies that have looked at genetically diverse populations, most only focus on a single layer of analysis. And what I mean by when I say that is your genetic diversity is all contained within your DNA, right? Your genome. But that genome is used to kind of control those further down processes because it's transcribed into RNA and then translated into proteins, which can perform cellular functions. And so looking at each of these layers individually can provide some information, but by combining, say, our genomic and our transcriptomic layers together, we're able to learn more than we would if we're looking at just any one layer by itself. And so that's kind of my goal for this experiment, is I want to combine both genomic and transcriptomic sequencing of these populations to see what we can learn both about the heritable variation itself, but also what we might be able to learn about potential regulatory interactions between these genes. And so when I'm talking about regulatory interactions, there's kind of two broad categories that we use to talk about the regulation of gene expression, which include trans-regulatory interactions and cis-regulatory interactions. Trans-regulatory interactions are diffusion mediated, so they can be between sequences that are very distant from each other in the genome, whereas cis-regulatory interactions are more allele specific, so they tend to be inherited, linked together, especially on the same cell. And specifically, I'm going to be looking for evidence of local cis-regulatory interactions because there's actually not a lot that we really understand about how the distances between these regulatory interactions can impact their function. And so I'm going to see if we can find evidence that these local regulatory interactions are playing an important role in aging. And so we use very similar methods to the previous chapter to sort our 4S population of yeast into 12 paired aged and young cohorts. And we'll then perform genomic and transcriptomic sequencing on each of these cohorts. And we're using pooled sequencing. So when we extract the DNA, rather than extracting DNA from each individual cell in each of the replicates, we're basically getting back measures of allele frequencies. So kind of estimates within each replicate of what proportion of alleles are present at each position of the genome. And then we're going to compare kind of the average sequences we observe between our aged and young cohorts to find regions of genetic differentiation or sequence level variation that are very consistent between aged and young cohorts. We can visualize this in what we call a Manhattan plot, where the x-axis represents position along the genome and the y-axis is a measure of statistical significance. So when we see points that are higher in our Manhattan plot, that kind of indicates that they're more likely to be important for this difference. And so these regions of high sequence differentiation can be represented as peaks. And so this is what my data actually looks like. As you can see, we do have a couple peaks that are sort of popping out of the data. Here, I'm looking at both SNPs and indels, or single nucleotide polymorphisms and indels, but I'm just going to refer to them both as variants and kind of lump them together. And I'm going to point out one variant in particular right here. This variant happens to belong to the TOR2 gene, which is kind of a famous aging gene. But what's more interesting about it from this standpoint is that the allele frequency trajectories that we observe, so the difference in allele frequencies between our aged and our young replicates, shift in a way that is very consistent between all 12 replicates. And so this is kind of a general feature of all of these highly significant variants within the gene. And that's exactly what we expect to observe with a polygenic trait, because there's no one gene we expect to have a large impact, but rather we expect there to be many genes throughout the genome that experience these small shifts in allele frequency to lead to changes we observe. And so as a whole, we identified over 70,000 variants, and 801 of those were significant. And of these significant variants, they corresponded to 132 unique genes. And so we took a look at this list of 132 genes, and we performed a GO-term analysis, or a gene ontology analysis of this list. And so essentially what a gene ontology analysis does is it pulls out different cellular processes, components, and functions that appear to be overrepresented in the genes that you're looking at in your list. And so it basically gives us an idea of which of these terms might have kind of a larger impact on this difference between our aged and young cohorts. And so we identified terms associated with the cell wall, cell membrane, or plasma membrane, and the cell periphery. Also, terms associated with glycosylation and glycoprotein synthesis and metabolism, and then terms associated with the Golgi apparatus. And so next, we wanted to take a look at gene expression. And so once again, we extracted the RNA from each of our cohort, or each of our replicates. And then now we're looking at expression level variation rather than sequence level variation. So we've already captured the sequence level variation, and we want to know how much of these transcripts are being produced. And we can visualize this in a heat map. This heat map is showing the 60 significant genes that we identified with our transcriptomic analysis. All you really need to know about this heat map is that all of our aged replicates are shown on the left-hand side, and all of our young replicates are shown on the right-hand side. And so you can see there's kind of this blocking in the heat map, which is kind of averaged out by this bar on the left-hand side, where we can see these top genes show increased expression in the aged, and these bottom genes show decreased expression in the aged individuals. And so I'm just going to point out two genes in particular from this heat map, and these are WSC4 and RFA3. And the reason these two genes are important is because they're actually the only two genes that were implicated in both the genomic and the transcriptomic analysis. And so these two genes are showing both sequence level and expression level variation between our aged and our young cohorts. And so as a whole, from these two genomic and transcriptomic analyses, we found evidence of several kind of known aging pathways that we mentioned earlier. The RFA3 gene that I mentioned is involved in DNA repair telomere protection, lots of genes involved in kind of glycoproteins, and also that TOR2 gene, the famous aging gene that I mentioned, is involved in dysregulation of nutrient synthesis. And so together, these kind of help to give us confidence that we're detecting true signals of aging, because these are all kind of well-established pathways in the literature. At the same time, we also uncovered some new insights that could be interesting. And so there's some genes like the SWE1 gene, which potentially could be involved in that asymmetrical partitioning of damaged materials. WSC4 was implicated in both of our data sets, and there's actually not very much known about that gene, so it could be interesting to learn more. And then WBP1 is actually the wheat germ agglutinin binding protein. And so you might remember from my introduction, the wheat germ agglutinin is the stain that we use to stain for butt stars. And so I'm going to come back to this result a little bit later, but I just wanted to point it out so that you know. But additionally, we found that there were only two genes that were indicated in both our genomic and our transcriptomic data sets. And so that's kind of an interesting result by itself, that it seems like sequence level variation is not really corresponding to the expression level variation that we're observing in these genes. And so we wanted to know what is. And so we took a look at all of those 801 significant variants from our genomic analysis and found that 58% of them were in genic regions, but 42% of them did not belong within genes. And that might not be surprising in cell systems, but in yeast, the majority of the genome is coding. So that's actually kind of surprising. And so we were wondering if those intergenic variants might be important regulatory variants. And so what I did, sorry, I skip ahead too early. So what I did was I looked for the location in the genome where all of these transcripts that were differentially expressed are coded. And I found the closest significant variant to each of these differentially expressed transcripts. So we're looking at the position within the genome where the closest variant is to each RNA transcript. And what we found was that the majority of these variants are not within 10 bases. And so we're not seeing many of these differentially expressed transcripts seem to show evidence of local cis-regulatory variation. And so as a whole, in this chapter, we were both able to identify genomic and transcriptomic patterns underlying these age-associated signals. And we also found that kind of this interesting result that there doesn't seem to be much evidence of local cis-regulatory interactions contributing to this variation. So it must be something else. And so finally, now that we have both established this reproductive trade-off and kind of found this genetic variation, we want to see if we can actually perform the selection experiment, right? Can we actually select on this existing variation to evolve populations of yeast that show these delayed immunogenic responses? And so to kind of return back to those Drosophila experiments that we talked about kind of at the very beginning, it's really kind of, the goal is to see if we can essentially replicate these experiments in our single cell system. And so there's a lot of kind of exciting potential opportunities to be able to do the same sort of thing in a different organismal system that we can easily manipulate and just generally show that it works in yeast as well as in flies. And so in fruit flies, the way that you can conduct these evolutionary experiments is you essentially just allow the flies to reproduce. And then any eggs that are laid early in life, you remove, whereas these eggs that are laid late in life, you allow to hatch and contribute to the next generation. And then over the course of several generations, this should improve their ability to reproduce in late life and, so forth. And we can do a very similar thing using methods we've already discussed in yeast. And so we can use cell sorting to separate out those high fluorescence aged cells. We then induce sporulation, so we have them sporulate, and we expect that our aged cells will show initially reduced sporulation efficiency because they're being forced to sporulate at late ages. Then we actually have these sporulated cells mate, and so any cells that don't mate, we actually kill, so that they really have to undergo the sexual reproduction at late age in order to contribute to the next generation. And so then we can just repeat that cycle continuously to essentially perform a very similar experiment to those that were done with flies. And so what we would expect to observe is that our control populations initially would have higher sporulation efficiencies than our evolved populations because they're not being forced to sporulate at late age, they're just sporulating, whereas our evolved populations have to sporulate at late age. We expect the control populations to maintain a relatively stable sporulation efficiency over time, whereas our evolved populations would need to improve their sporulation efficiency in order to adapt to these selection conditions. Unfortunately, or I guess maybe not unfortunately, but that's not what we observed. And so what we saw in our data instead was both our control and evolved populations showed a steady decrease in sporulation efficiency over the course of the experiment. And there wasn't really a clear difference between those control and evolved populations either. And one thing that's not immediately obvious, just looking at this graph by itself, is that these sporulation efficiencies as a whole are actually much lower than they were in our first experiment, where we were looking at those trade-offs. So 20% sporulation efficiency would have been very low in that first experiment, And we only have one template that achieves a sporulation efficiency greater than 20% in this experiment. So as a whole, there's something going on here where we're seeing this really reduced sporulation efficiency compared to what we've seen previously. And so one potential explanation for what might be causing this is a genetic bottleneck. And so it's possible that somewhere along the line, we lost a lot of the genetic diversity that these populations can kind of use to adapt to these conditions. And natural selection can't act on genetic diversity that isn't there. And so if we have kind of the effects of this bottleneck, it could lead to these kind of depleted sporulation efficiencies. And so we wanted to kind of take a look and see if we could find evidence of that. And so we looked first kind of generally at our genomic data. And in this Manhattan plot, we actually don't have any significant genetic variants. They're all below that significant threshold. We do, however, see evidence that there are kind of these small peaks within the data. So we wanted to look a little bit more closely at those. And we did find, when we looked at our allele frequencies, that just like in the previous experiment, these peaks do seem to be driven by these kind of consistent shifts in allele frequencies that diverge between the two different groups. And so even though we're not seeing any significant variance, this does look like it could be an indication that there's some weak selection taking place. And so we looked a little bit more closely at genetic diversity itself by using an estimate of heterozygosity. So heterozygosity is essentially, just an estimate of the genetic diversity available within a population. And a heterozygosity of value of 0.5 is very high, whereas a heterozygosity value of 0 is very low. That's what you would see in a isogenic population. And so we looked at our genome-wide averages for heterozygosity, both for our ancestral population and then for all 3 cycles of our experiment for both the control and evolved treatments. And what we found was that there does seem to be a pretty steep decline in heterozygosity between our ancestral population and the first cycle of our experiment. So it does seem like there's evidence that there may have been a genetic bottleneck that occurred very early on in this experiment. Additionally, we kind of, to visualize this, I zoomed in here on chromosome 5, and then this kind of gray rectangle within this plot of chromosome 5 indicates that peak within chromosome 5. And so you can see in this figure that while heterozygosity decreases pretty substantially between the ancestor and the first cycle of the experiment, it remains pretty constant throughout the next few cycles, and it's also very constant between our evolved and our treatment populations. So it seems like the effect is pretty even between our 2 treatment groups, with the exception being these peak regions, where we see, much like in the allele frequencies, kind of this divergence between our control and our evolved populations. And so given that this is only occurring within these same peak regions, it seems like it might also be another indicator that this is a result of like purifying selection or some other natural selection reducing that heterozygosity rather than genetic drift. And so given that evidence that we may have seen some weak selection in our genome, even though we didn't have any significant variants, we decided to take a look at the 1% of most differentiated variants within our genome, just to kind of see what was in there. And so we performed another GO term analysis and found, once again, that terms associated with glycosylation and glycoproteins were frequently represented. And another protein modification, both from protein kinases and phosphatases, where phosphate groups are added and removed from proteins, was also identified, in addition to just general regulation of transport. And so together, we found from both this experiment and the previous experiment that protein modifications seem to kind of frequently be popping up in our data, especially these glycoproteins and like this TOR2 famous aging gene and some of the other protein kinases. So that's kind of an interesting repeatable result from both experiments. In addition to this WBP1 gene, which also showed up in both experiments that I promised we would talk about later. And so what's kind of interesting about the WBP1 gene is that since it's binding to the stain that we're using to sort our cells into aged and young fractions, on one hand, it's kind of cool because it's showing that our methodology is kind of directly related to the genomic results we're getting. So it does seem like these genomic signals are a result of selection, right? We're identifying genomic signals associated with selection on our sorting procedure. However, it also kind of raises the question of which of these signals are directly associated with aging and which may be associated with kind of other technical artifacts of our data. For example, maybe this protein has better binding affinity or something like that. And so we need to do a little bit more work to kind of tease apart which signals within our data are age-associated signals versus what might be technological artifacts. Luckily, though, cell sorting isn't the only method, or I'm sorry, like fluorescence activated cell sorting isn't the only method of sorting cells and kind of doing this age differentiation. And so there's a lot of opportunities that we can use to sort cells using different methods and all of that to kind of get at this question of tearing apart those different genomic signals. And so today, as a whole, in order to kind of address this question of how evolution shapes age-related variation, we've gone through these 3 experiments that are both trying to kind of establish Saccharomyces cerevisiae as this exciting system that we can use for the experimental evolution of aging, and also take a look at these kind of broader processes that control aging. And so we found that recombinant population of Saccharomyces cerevisiae does show age-associated trade-offs expected under evolutionary theory, establish that there's genomic variation that seems to underlie these shifts or these differences that we see. And we have also shown that we can select on these phenotypes that have consistent shifts that we would expect in aged cells. And so as a whole, we're really excited about this system and kind of the future opportunities that it provides for us to be able to look at kind of the genomic basis of aging and possibly other traits as well. And so, okay. And so I want to go through my acknowledgements. And I'll start out by saying thank you to my funding sources, Oregon State University, the NIH, and also the Collins Medical Trust, which have provided all the funding to make this work possible. I also want to give a big shout out to Julia, who was an undergrad SURE student who helped with the selection experiment. And she did a lot of work that unfortunately didn't make it into my slides, but was like completely critical for us to be able to actually do that selection experiment. So a huge shout out to Julia for all of her help. I also want to say thank you to Paul and Mary Ann Roberts and also the Oregon Moderates Fellowship. I'm very, very grateful to have received support from them. And a huge shout out to my lab for just making doing science a lot more fun and always being there to help with coding support and offer advice on manuscripts and talks. And also shout out to Bryson, who took some of the photos that you really can't see very well, but were the background for a lot of my slides. I also want to say thank you to my committee members who are just really amazing people, and I'm really, really grateful to have had them on my committee. Chris kind of introduced me to the fact that yeast are also organisms and not just bags of yeast, as Molly once said. I took a class with him, I think my very first term here as a grad student before COVID hit, and got to learn a lot about the ecology of yeast, so that was really exciting. And Virginia, I think it's funny because I think the reason I originally added you to my committee didn't really end up being that much, but I am so, so grateful to have you on my committee because you have just been amazing to have in all my committee meetings and just a very supportive person. So I'm really, really grateful to have you. And Erin also, I had a terrible time finding a GCR and then eventually just kind of threw away the GCR list and emailed Erin because I was like, he seems like he does cool stuff. And he was so, so kind to be on my committee. And I'm just really, really grateful to have you on my committee as well. And of course, Felipe has provided so much help with, not just help, I mean, Felipe like helped design the second chapter of my dissertation and has just been like so great with designing and writing that manuscript, and it's so much better for having his involvement. So yeah, thank you to all of you. Also, Molly deserves slides all to herself. I really couldn't have asked for a better PhD advisor. The best advice I got when I was applying to grad school was from my dad. Shout out to my dad. And he basically said your advisor is going to make your right for your grad school career. So make sure you have a good one. And yeah, I couldn't be luckier. I'm just really, really fortunate to have you. Also a really big shout out to kind of the greater Mercado Lab ecosystem. It's been really great to have your community as well, especially when I was the only grad student in Molly's lab. It was very, very nice to be included. And it's been great to kind of share. Okay. Yeah. To share like the experiences of going to conferences and all that. And I also want to say thank you so much to the office staff at Integrative Biology, Tara, Melissa, Tracy, Teresa, and Rowan. Having you around just like helps the world continue to function. I can't tell you the number of times I've emailed one of them and just been like, I have no idea what to do. And they just swoop in and help. So it's, yeah, we just try to function without you. And also a huge shout out to so many of my teaching mentors. And Molly falls under this category as well. But I just learned so much when I've been here, and I've had so many great opportunities from Lori and Carmen and Sloshina and Adam and so many other people to kind of build my teaching portfolio and learn what I really like about teaching. And so that's been amazing, and I'm really grateful for those opportunities. And then of course to just the greater IB community as a whole. It's been amazing to be here, and I'm just so grateful that we have such a supportive and awesome community. And yeah, so you all know who you are. And yeah, I'm very, very grateful to have been here with you. We're almost done, I promise. Okay. And then finally, a huge shout out to my family. I'm just completely floored by how many of you came all the way out here in Oregon today, and how many of you are watching over Zoom. So I'm really, really grateful to have had you around to kind of help me maintain my identity as a human outside of grad school too. And so yeah, just really, really grateful in general. And now I have time for questions, I think, if anybody would like to ask.
Like, what's your separation between young and aged cells? Like, is there a certain number of buzz cars, or is it just amount of fluorescence?
Yeah, it's a great question. That took kind of a bit of trial and error. So kind of the first, like prior to this experiment, I did quite a few sorts where I was just kind of playing around with different fluorescence thresholds and trying to see what would work. And so in the end, it was just kind of like a bit of a judgment call on this seems to be doing a good job. But if you push the threshold too far, then most of your cells die. And then if it's not far enough, you don't really see any difference. And so, yeah, it was just kind of some trial and error of seeing like, okay, how far can we push it? Or we still have enough living cells that we can actually do this experiment.
Yeah. I might have missed it because I came in this late, but is the rate of budding linearly related to age? Like calendar age?
So like time, you mean?
Yeah, I guess I'm just curious if budding is reflecting also maybe kind of pace of life or metabolic traits.
Yeah.
In addition to actual calendar age, whether or not, I don't know, I'm not even sure that actually matters in the concept of aging, but what do you think?
Yeah, so like on like the genetic basis, there's a lot of overlap between like chronological age and replicative age and kind of how those interact with each other. In terms of, it kind of depends on like if you just have some yeast like hanging out in culture, they tend to replicate at a pretty like regular, like about every 2 hours ish. But if there's different conditions that can kind of change, like you can just freeze them and they just won't replicate at all. And so it kind of there's definitely relationships between chronological age and a replicative age, but they can also kind of be separated from one another in some ways.
Yeah, so I have a question about chapter 2. So I think the lack of concordance between the genomics and transcriptomics, that's like pretty surprising. Are you sort of interpreting that biologically as there's a lot of transregulation, or do you think there are like technical features of like something else that can be driving that?
So I think that's a great question, and I'm going to quickly go on a tangent, but I'm going to come right back. So I've actually gotten both ways. I've had people say, well, that's not surprising at all. Like, why would you care? So I think it's interesting in that perspective that people have such different ideas. But I think, yeah, I think transregulation is definitely one of the possible explanations. But also we could have just more distant kind of cis regulatory interactions that we weren't able to look at. And there could also be, I mean, I guess it depends on like how broadly you define trans regulation too, because there's also, you know, all sorts of other things we could do kind of farther down the cascade to break down RNA or that sort of thing. And so I've read kind of a lot of different definitions of how broadly people define trans regulation. But yeah, I think that's definitely like one really good hypothesis for what could be going on.
Well, if we don't have any more questions, we can conclude this part of the defense, and then the committee will convene to our closed door session for a few hours to grill Katie. But let's thank Katie for her talk.